sugars within our tissues. And when I started my lab back at Arizona State, we were interested in the role these long-chain sugars played in the mechanical properties of tissues. But it wasn't long before I realized that most of the long-chain sugars are attached to a protein core. And it's the combination of the protein core and the long-chain sugar that give rise to the properties of the materials. And so we've made several mimetics of the proteoglycans. They're very hard to isolate from tissues because they're there in small quantities. And unlike just a protein, it's really hard to make them in bacteria or yeast because they're post-translationally modified, and the bacteria and yeast don't do that the same way that human cells do. So we tried to come up with synthetic methods to try to recapitulate much of the function of these proteoglycan mimetics. So we made a decromimetic and looked at that in wound healing. I was going to talk about that today, but I'm not. And we've made a mimetic that I'll talk about today that mimics the surface of the endothelium, the healthy endothelium and blood vessels. Then we've made some for cartilage, aggrecan and lubricant. I won't talk about those at all, but but you can look for papers on those if you're interested. So one of the drivers of my laboratory is inflammation, because inflammation plays a role in many different disease states, and in fact in most diseases. If anyone wants to come sit closer, there are chairs up here. I feel bad that everyone's standing in the back of the room. But inflammation plays a role in many diseases and in tissue healing, and it's really important in tissue healing, so you need some inflammation. But when it goes awry, then you end up with things like fibrosis in different tissues of the body, or some level of chronic inflammation, and what like you see in metabolic syndrome that people with diabetes and others have. And so we're trying to find a way to steer the body back to the gentle inflammation at the beginning for tissue healing, and then for that inflammatory pathway to die down. So in what I'll talk to you about today, what I mentioned, we're really interested in the endothelium. And on the right side of this slide is a transmission electron micrograph of the inside of a blood vessel. And just at the interior of the blood vessel are the endothelial cells. And they have this thick glycosaminoglycan coating called the glycocalyx that really looks like this hairy structure. And it sticks up from the surface of the cell, and it essentially masks all of the receptors on the surface of the endothelial cells. And that helps control the interaction between blood cells within the blood and that endothelium. So in a healthy blood vessel, the cells in the blood kind of just flow by. But if the endothelium is damaged, then those inflammatory cells and platelets can interact with the surface of the cell. So that's what we see here. There's now a transmission electron micrograph of a damaged endothelium, and you see that hair structure is gone. And now there are receptors that are exposed on the surface of the cells. And that allows things like neutrophils and platelets and things to interact with that blood vessel surface. And that leads to thrombus formation or soft clots within the blood vessel, as well as inflammation. So we wanted to come up with and this is, I'm sorry, I thought it was on the next slide. And this is just a cartoon of this where you see there are E and P selectin receptors. There's the receptors for growth factors. And these selectin receptors will start to capture some of the inflammatory cells, and then there are integrin receptors that also play a role in really allowing the inflammatory cells to migrate into the underlying tissue and cause fibrosis and other things. So we didn't feel like we wanted to stop the entire inflammatory process, so you need the glycocalyx to disappear, and you need some inflammatory cells to interact with the cell surface. But we wanted a way to dampen that interaction, so you didn't have exuberant inflammation. So the first thing we said was, well, what is this glycocalyx composed of? So I mentioned that there's a protein core. In this case, the protein is often embedded into the cell membrane, and then there are glycosaminoglycans that come off the protein core. One of those glycosaminoglycans is called dermatan sulfate, and we're very interested in dermatan sulfate. It's not as anticoagulative as heparin, which is used in surgery to anticoagulate people, but it has a little bit of anticoagulant property, and we wanted to be able to use that to try to be a little bit antiplatelet to stop some of the coagulation on the blood vessel surface. So we started with dermatan sulfate. And just to remind you what we're trying to do. So here's our healthy endothelium on the left with a glycocalyx. When there's inflammation, that glycocalyx disappears and you have receptors exposed on the surface of the endothelium. And we're trying to create essentially a Band-Aid to try to limit the cells that interact with those damaged endothelium. So in a healthy endothelium, the cells just flow by. And in the inflamed environment, many of the inflammatory cells will bind. We're trying to get rid of that. Okay. So I'm going to call our glycosaminoglycan peptide conjugate, or our proteoglycan mimetic, two different things. The beginning, it's ECCl, so that's variant number 1. I was thinking like a sealant for the endothelium when I named it. And then the second, when we've improved it, it's called DSIKL, and that's dermatan sulfate and IKL are the first 3 amino acids of the peptide that we've used. All right, so James Wadika, who was an MD PhD student in my lab, he's now a cardiology fellow at Emory, designed the first molecule, and he took dermatan sulfate, he oxidized it, so you can see the ring structure opens up and there are aldehyde groups, and I bet this is animated. Yep. And then he could put a hetero-bifunctional cross-linker on the molecule called BMPH, and that allows us to, add the peptide handle. The amino acid sequence is shown here, and again, conjugated to dermatan sulfate. And then he did a lot of work to show the peptide was conjugated, the molecule was what we thought it was, a lot of other in vitro work. And then he started working with Vasilios Maroukas, who is now a postdoc at Washington University in St. Louis, and he was in Scott Simon's lab at UC Davis. And so we worked with that lab to start to understand whether we could inhibit neutrophil interactions with endothelium under flow. And so they put endothelial cells down on the surface, they'd inflame the endothelium, and then they would isolate human neutrophils, activate the neutrophils, and allow them to flow over the surface. And if we have neutrophils rolling over the surface of activated endothelium, you see a lot of neutrophils will slow down in their rolling process and stop on the surface and try to migrate through that endothelium. If we treat with our molecule, we reduce the number of neutrophils that slow down and those that are trying to migrate through. And it works similarly to a pan-selectin inhibitor that's a small molecule. We tested it, was in human clinical trials at the time. In vivo, our molecule should work better than the small molecule, because the small molecule will bind one receptor. So if it comes off, it flows away. Ours binds multiple receptors at the same time, so only some of the peptide will release, and so it stays bound better. So in theory, it should work better over time. This is just a snapshot in time. Okay, so we knew the molecule, we could make it. It bound to inflamed endothelium and seemed to inhibit inflammatory cell binding to the surface. And so we moved very quickly into an animal model of deep vein thrombosis. And this animal model was developed by Tim Wakefield's lab at University of Michigan, and my colleague at Purdue, Craig Gorgon, recapitulated or essentially mimicked the model in his lab. And you essentially ligate the inferior vena cava in a mouse, and you ligate it so there's only about 10% of the flow that there normally would be. And when you do that, the blood vessel expands dramatically, and you get poor blood flow and eddy formation where the blood vessels expanded. And this allows for thrombus formation to happen very quickly. And it's very similar to what you see in humans with respect to the cell biology when humans have deep vein thrombosis. And the nice thing about this model is you can read out in about an hour. So it's a very fast model. So we used 6-week-old mice, we ligated the inferior vena cava, and then we dosed the tail vein injection about 20 minutes after that ligation, and used ultrasound to look at thrombus formation. And we used heparin as a control because heparin should systemically anticoagulate the animal. And if you look using ultrasound, what you see is after 6 hours, our molecule and heparin both work fairly well at reducing the degree of thrombosis. The black bar is if there's no treatment. But after 24 hours, what we found was heparin is still working pretty well, but our molecule is not working as well. So there was a little bit of concern about half-life there, and we'll get to that. That's why we have the redesign of the molecule. But I want to try to convince you that while heparin systemically anticoagulates an animal, our molecule must be working locally. So we did some coagulation tests, and what we found is that as you increase the amount of heparin in blood, you slow down the coagulation time. So you inhibit coagulation of the blood. You can add our molecule, and at very, very high concentrations we dosed here, you do not anticoagulate the animal. So if we're inhibiting thrombosis, it must be acting locally and not through systemic anticoagulation like heparin is. Okay, so there are a couple reasons why our molecule might have a shorter half-life than we would like. So the amino acid sequence we took originally was from phage display, and it's all L-amino acids, which are the normal amino acids, and we all know that proteins are subject to proteolytic degradation. And so we thought maybe the peptide's being chewed up by the proteases that are available in the inflamed environment. And the other thing is that in our original chemistry, James oxidized the glycosaminoglycan to open up the ring structure to add the peptide. And that actually makes the molecule much more susceptible to hydrolysis, and so it also can degrade more quickly in this inflamed environment. So we wanted to change two things. We wanted to try to change the peptide to incorporate D amino acids, so it would be less susceptible to enzymatic degradation, and change the chemistry. So let me talk to you about the peptide first. Okay. So this work is Tima Dagani's work. She now works at In Citro, which is a AI company focused on more rapid drug discovery in the Bay Area, out in San Francisco. But she used a process that was developed by Kit Liam when he was a postdoc. And Kit Liam's a faculty member out at UC Davis, so she worked in his lab to do this. But it's a combinatorial approach. So let's talk through some solid-state peptide synthesis. So when you synthesize a peptide using chemistry, we use beads. And so that's shown by these sort of dark circles here. And each bead has thousands of the same peptide growing off the bead during synthesis. So each bead has a unique peptide, but thousands of copies of it. And normally in peptide synthesis, you have all of the beads together, and so you're growing thousands of peptide chains that are identical on all of the beads. But in Kit's process, what he did was he takes the beads and puts them in X number of different pots. In this example, it's 3. And he adds an alanine to the first one, for example, glycine to the second pot, and valine to the third pot. And so now all of the beads in pot A have alanine, all in pot B have glycine, all in pot C have valine. You mix them all together, and you split them out again. So now the first amino acid is either A, G, or V. If you add an alanine, the second has to be alanine. Similarly here, it's A, G, or V, but the second amino acid is glycine. Here, the second amino acid is valine. You mix them all together again, and you start your next addition. And so you end up with millions of beads, and each bead has a unique sequence, but it has thousands of copies of that unique sequence. And so this is what Tima did. She built out a library. You get a huge number of unique peptide sequences. It's very easy to incorporate unnatural amino acids because we're using chemical synthesis outside the body, and they're very simple screening methods. So she takes the beads and she glues them down to a plate, and then you can use just simple like an ELISA test. So she takes an antibody to E-selectin, tries to bind it to the different beads, and then uses a colorimetric assay to see which beads have this antibody. And any bead that turns brown has the antibody, so you can take that bead and then sequence the amino acid sequence on that bead. And then you can start to understand what amino acids are important in each location. So in doing that, she found a new sequence that bound very well to E-selectin, and it was selective for E-selectin. So it didn't bind to ICAM or VCAM. It bound a little bit to E-selectin, but not well. And that is arginine, hydroxyproline, D-proline, leucine, D-lysine, and isoleucine. Okay, so that is how she found her new peptide. For the chemical conjugation, remember we used to open up the ring structure where the 2 hydroxyl groups were. Now she's looking at the carboxylic acid group. She's using a molecule called DMTMM, which is very similar to carbodiamide chemistry to activate the carboxylic acid group. And And then her peptide has a hydrazide component, so she can conjugate the peptide. Whoops, and so we end up with this molecule DSIKL, and for each dermatan sulfate backbone, there are about 15 peptides conjugated to it. Okay, so just to remind you what we're doing, remember we're trying to recapitulate the glycocalyx, right? So the endothelium, it's inflamed, it's lost its glycocalyx, so we're trying to form this band-aid that'll bind to the endothelium. Going backwards. Okay, so at this point, Tiemann did all of the same characterization that James had done. We know the molecule binds to E-selectin. It binds a little bit to P-selectin. That's probably from the dermatan sulfate, because dermatan sulfate is known to bind to P-selectin. It's another one of the inflammatory receptors on the endothelium. Doesn't bind to ICAM or to VCAM, all of those kinds of things. And then we moved into a microfluidic system that was developed in Steve George's lab. So in this system, we have outer channels. They can be filled with saline, with gel, or with a tissue of interest. So at the beginning, it's just going to be a gel. We have endothelial cells on the inside. Those are shown in red. We stain neutrophils that we isolate from humans in blue. And then there are these little porous structures where cells can migrate through the endothelium into the underlying tissue. Okay, so if we line the system with endothelium, we can inflame it with TNF-alpha and IL-1 beta at concentrations that are found in patients with COVID. And then we can either treat with our molecule at increasing concentrations, not treat with our molecule and flow neutrophils over. So if we don't treat with our molecule and we flow neutrophils over, you can see neutrophils bound to the channel, and you can even see neutrophils migrating through the endothelium into the outer gel. Okay? And that decreases with increasing concentrations of our molecule. You see fewer neutrophils in the surrounding gel and bound to the endothelium, and that's quantified here in both cases. And just to show you what this looks like, here if I can find my mouse, yeah, okay. So when we don't treat with our molecule, but we inflame the endothelium, you see the neutrophils are moving very slowly across the surface. If we do treat with our molecule, the neutrophils move pretty quickly across the surface, right? So they are binding to the endothelium. All right, so I get this to stop so I can move forward, maybe. Okay, all right, so the next thing we wanted to know is that we can decrease the neutrophil interaction with the endothelium. How does that affect underlying tissue? So in Steve George's lab, they use human-induced pluripotent stem cells and convert them to cardiomyocytes. So those are now tissues in the outer chamber. There are also some cardiac fibroblasts to stabilize the cardiomyocytes. We have our endothelium and our neutrophils. Okay. And, well, yeah, okay. And so again, we're going to stimulate with inflammatory molecules. So if we stimulate with TNF-alpha and IL-1 beta, we don't have our molecule and we don't have neutrophils, we can measure a beat rate, and that's the beat rate. So the happy cardiomyocytes are beating at some normalized rate. If we add inflammatory agents, that beat rate does not change. If we add inflammatory agents and our molecule, the beat rate still doesn't change. However, if we add inflammatory agents and neutrophils, the beat rate dramatically increases. But if our molecule is there, it goes back to normal. And it's known that neutrophils will affect heart rate, and there are arrhythmias in some COVID-19 patients. And so this is a way to start to understand what's causing the arrhythmias and are the neutrophils involved. What does not happen is, you know, in like a neuron, there's a rate at which the action potential goes across the axon. It doesn't affect the rate that the action potential travels in the cardiomyocytes, it just affects the rate of heating. So, when this project started, all the way back with James Wadicka, I was interested in two things, one of which was in ischemia reperfusion for heart attacks. So, many of you likely know someone who's had a heart attack and survived. or someone who has diseased coronary arteries where the physicians want to go in and use a balloon to reopen that artery and put a stent in to try to reperfuse the tissue. So when you do this, you go in and you open the coronary artery and you reperfuse the tissue. Ironically, that reperfusion causes additional damage to the tissue, likely because of inflammation and neutrophils and other things migrating into the underlying tissue and causing fibrosis. So we wanted to ask the question, if we delivered this molecule just after reperfusion, could we limit the amount of additional damage to the heart tissue? Okay, so to do this, we worked with Panitch and Binodat's lab at UC Davis, and this is Feng Tai, who is an amazing microsurgeon. So he went into these little mice, and he tied off the left anterior descending carotid artery, and he tied it off for 40 minutes, and then he opened it back up and allowed reperfusion. And after reperfusion, probably 15 minutes or so, because they sewed the incision back up, they dosed with either saline, so just a salt solution, or with our molecule via tailmate injection. And then we looked at outcomes at 24, 36, and 2 weeks. Okay. So the first thing we wanted to know was, are we actually targeting the heart if we deliver butyl-bane? And so we labeled our molecule with red dye, and we used in vivo fluorescence imaging. And if you do that, you can see that there's really dark imaging right around where the heart is. There's actually lots of color in the stomach too that the vets think is because of the food that the animals were eating. And then you can see where the animal was intubated, where there's also some endothelial damage. But in animals that are treated with saline, you don't see any of that. So likely we are targeting the heart. And if we look at and quantify the amount of fluorescence in the heart at 1 hour, so these are sham animals, they were opened up, there was no ligation done, they're treated with our molecular saline, you don't see any targeting to the heart. In animals that were injured, and treated with saline, you don't see targeting to the heart, there's no molecule. This is our molecule that's labeled, so you see it at 1 hour, at 12 hours, at 24 hours, and we redosed. If we had analyzed the data first, we would not have redosed, because we would have wanted to know how stable is the molecule, right? That was one of our questions. I think it's more stable because it's still there at 24 hours, whereas in deep vein thrombosis, we lost activity at 24 hours, but we don't know for sure because we redosed, but the molecule is there at 36 hours. So that was good news. We also isolated the coronary tissue at 24 hours, and we used a TTC dye, and with this dye, if you have white, it suggests that you have an infarct region or a region where there's no blood perfusion. And if you quantify that region, what we find is if we treated the animals with saline, there was a larger infarct than if we treated them with our molecule. So that was good news. We have less tissue that seems to not be perfused. And we followed that up by taking blood from the animals and looking for eye troponin, which is a marker of cardiac damage, and there's less eye troponin in our treated animals. If we then isolate all of the cells from the heart and look to see the distribution of cells, and our paper shows all of the different cell types we looked at. I'm just going to show neutrophils because that's been sort of the line of the story I've been telling you. In sham animals, around 1.5% of the total cell population are neutrophils, as determined by flow cytometry. In saline treated but injured animals, that goes up to almost 3%. And if we treat with our molecule, it comes back down to almost normal levels. And that is quantified over here. So these are sham control animals, no difference. Saline treated versus our molecule treated in injured animals. We see that decrease in neutrophils. And this is in agreement with what we see with immunohistochemistry at 2 weeks. So if we look at the actual damaged tissue and we look for macrophages or neutrophils, what we find in sham animals, we don't really see macrophages or neutrophils in the tissue, which is expected, it's not damaged. In damaged tissue that we treat with saline, we see both macrophages and neutrophils at 2 weeks. And in the treated animals, we don't see much of either, and that's quantified on the right. And if we then look at function, we can look at the ability of the heart to contract, that's fractional shortening, and we see improved ability of the heart to contract with treatment. We can look at the mitral valve deceleration time, which is indication of ventricle function, and we see improved ventricle function. And we can look at diastolic function, or the ability of the heart to refill early and late and diastole, and we see improvement in the ability of the heart to fill with blood. All right. And I just want to remind you that this is likely due to a local effect. If we look at the ability of heparin to anticoagulate blood, it anticoagulates at very low concentrations. Dermatan sulfate itself anticoagulates at much higher concentrations, and our modified dermatan sulfate anticoagulates at even higher concentrations, and we're treating down in this range. So we can't be systemically anticoagulating, suggesting it's a local effect. All right, so in conclusions, I hope that I've convinced you that we can use chemistry and some, what I hope is intelligent design of molecules to design something that can recapitulate the glycocalyx of the endothelial cells so that we can dampen the ability of some of the inflammatory cells like platelets and neutrophils to interact with the endothelium and thereby improve tissue healing. I didn't directly show you the evidence of fibrosis as I mentioned. Well, I guess the TTC staining showing all the smaller infarct area suggests that. The paper does look at activated fibroblasts and the histology as well, so we also reduce fibrosis. I think I mentioned everyone who worked on the project as I went through, but these are the folks who worked on the project. These are people from Steve Turner's lab, whose names I did not mention. People in my lab who did a lot of the work, and then the funding agencies for this part of the talk. Okay, so I'm going to completely change gears on you and talk about nanoparticles for drug delivery. And I'm going to try to talk to you about the general design of the types of nanoparticles that I use, and then give an example in breast cancer. All right. So, for many years now, my lab has worked on responsive, degradable nanoparticles, and they're hollow nanoparticles, and we'll talk about that in a minute. And this is actually a project that I adopted because one of our faculty members at Purdue started working on this and decided that academia was for the birds and he was going to law school and become a patent attorney. And I took in all of the students who were working on this nanoparticle project, and I thought it would go away, and we're still doing it. It's like 15 years later. But okay, so we're working with these polynithium particles. And polynithium's a really interesting polymer. So it's a little counterintuitive. It's thermoresponsive, and as you cool it down, it will go into solution, and as you heat it up, it comes out of solution, or it becomes like a polymer-rich phase. So it's the opposite of what you would expect, right? But so what's interesting is you can cross-link it, and we form these little particles, and if we cool them down, they expand. They can't go into solution because they're cross-linked, right? But they'll expand, and then when we warm them up, they'll collapse. So that's really useful for loading biological molecules into the materials because we can load at cool temperatures, and then they'll collapse around body temperature, and you can control the release because now they're smaller, and the pores are smaller, and things diffuse out more slowly. Okay, so we have the polynithium, or the nithium monomer. We use another acrylated monomer that has a sulfonic acid, so it has a sulfate group to mimic the sulfates in glycosaminoglycan, so there is a theme. We use acrylic acid because it has an acidic group that we can use for chemistry, so we can put peptides on them. And we use a degradable crosslinker that has a disulfide bond, so that the crosslinker can fall apart in the body over time, right? So the nanoparticles fall apart and degrade. Okay. Let's see. So I mentioned that they're hollow, and and this is intentional. We did a ton of work with these polymers over the past several years. And what we found is if we didn't first make a core and then get rid of the core, the polymer was so dense we couldn't get all of the peptide or whatever we were delivering out of the polymer. So to fix that, we make a core. We make the core by just taking the NiPAM polymer and no crosslinker. We make really low molecular weight polymer, and we make these tiny little cores. We stop the reaction and then restart it by adding all of the monomers we want to have in the particle, including our disulfide crosslinker. So now there's a shell around it that's cross-linked, and we can cool it down, it'll swell, and that allows the core to diffuse out. So we get rid of all of the initial polymer, and that makes it pretty low density in the center and allows us to deliver molecule. So we end up with this degradable shell that, again, we often call hollow. It's not really hollow, it's low polymer density. Yeah, okay, so those are the shells only. And then I mentioned we have acrylic acid, so we can modify with targeting ligand, and so we can also conjugate peptides to the surface. We often use a collagen binding peptide. And so we have these nanoparticles that are fairly low polymer density. Remember, we can polymerize around a core. We can load and release some kind of drug, and we can target. Okay. So, I've been working with a woman, Luciana Lopez, who I'll mention again in just a couple minutes, who is at the University of Sao Paulo in Brazil. And Luciana, when she was a graduate student, visited Colleen Brophy's in my laboratory at ASU for a year during her PhD. And then she came back and did a postdoc with us, and then she went off to Brazil and started her own lab. And several years later, she called me and said, "Hey, Alyssa, I have this great graduate student, can I send her to your lab for a year?" And so at that point, we started working together. So she was really interested in breast cancer, and in particular in ductal carcinoma in situ. And so these breast cancers start within the milk duct, and then they can become invasive and start to metastasize out of the milk duct. There have been lots of long-term studies, and what they show is about 20 to 50% of individuals who develop ductal carcinoma in situ progress to this more aggressive cancer, but the rest of the people never progress. Yet, we still treat people with chemotherapeutics, right, that are systemic drugs that are toxic to the body, with radiation that can cause lung fibrosis and other issues, and with long-term medications. So, more recently, there have been lots of questions about whether they should be treating these people who have low-risk carcinomas with these kinds of chemotherapeutics. And there have been some studies showing that you can actually inject therapeutics into the milk duct and get local effect, and that maybe that kind of approach is a better approach for this type of cancer. And so this is what Luciana's group wanted to do. I'm going to tell you the two vignettes in this in the opposite timeline in which they happen, because I think they make a little more sense this way. So Luciana, as I mentioned, I've known her for many, many years since she was a graduate student. She's now sending her graduate students to our lab. Julia Passos is the graduate student who came to work on this project, and she had developed solid lipid nanoparticles. So the particles are made out of lipids. She could encapsulate small molecule hydrophobic drugs within the lipids, and she was using them for introductory administration. But she knew that we could do collagen targeting by putting our shell, our NiPAM shell, around things. And she wanted to know, could we put a NiPAM shell around her solid lipid nanoparticle in order to target to the tumor itself? So we could do local drug delivery. So this is what she wanted to do. She had her lipid nanoparticles that she made in Brazil, and she wanted to put our NiPAM shell around them. So it sounds like a really easy task, right? We've already shown that we can take cores and put the shell around them. It was a little trickier though, because the lipids melt, right? So butter is made of a lot of lipids, right? You think about your exposed heat, it melts. Well, our NiPAM initiator to start the first thing that Julia had to do was say how much heat can she use, or to what temperature can she heat up her lipid cores without them melting. And what we found was really 50 degrees was ideal. They start to melt at 60, but they really melt at 70, so we can't go there. As she puts her drug in there, you see this a little bit more, right? They're a little less stable even at 60 degrees, and certainly unstable at 70 degrees, although ironically a little more stable than they were without the drug. And so then she said, okay, well, at what temperature can I make the polynipam? So what she found was, do this right. So this is the nipam core with the shell around it, made at 50 degrees. This was the core with the shell around it at 60 degrees, and this is the core and the shell around it at 70 degrees. And you see that here, oh boy, I'm backwards on this. Hold on, let me come over here. So then I think the data is backwards. Regardless, what this showed was she couldn't form the shell. You can see that there's the particles get larger here. Oh yeah, no, this is right. So the particles get larger at 70 degrees. They get larger at 60 degrees, but not as well as if you polymerize at 70, and at 50 degrees you can't form a shell at all. Sorry. My brain was completely blank for a minute there. Okay, so we have to use at least 60 degrees because we can't form the particles if we're below 60 degrees. And we can't go higher than 60 degrees because we melt the lipid cores. All right, so 60 was the optimal temperature to move forward. Phew. Okay, so then what we wanted to know was, are we really forming a shell around the core, right? Because it's quite possible that we put the cores in and we initiate polymerization and we form nipam particles and we have lipid cores and they are completely separate entities. And so we labeled the lipid core yellow and we labeled the nipam red by incorporating a monomer into the backbone of the polymer. And we used flow cytometry that you usually use for cells, but you can use for fluorescent nanoparticles. And we said if the shell has formed around the core, we should find overlapping fluorescence, right? Yellow and red fluorescence. So in flow, if you have an unlabeled particle, you find it in the lower left-hand corner. If we have something that is labeled with yellow or green, we're going to find it in the lower right-hand corner. If we have something that's labeled in red, it's in the upper left-hand corner. And if we have, in theory, something that's the core with the shell has both overlapping, you'll find it in the upper right-hand. So in her particles, when she purified them once, we found there's some just cores, and there's some dual, core, shell around a core. And if we purify them again, we find all shell around the core. So we have a nice purified core shell nanoparticle. Okay, so she also wanted to look at loading efficiency. So for her solid lipid nanoparticle, she had really nice loading efficiency, right around 93%. But when she heated them up for polymerization, that dropped to 75%, so she lost some of the drug. If she took the polynipam nanoparticle, so she formed just the particle that I talked about at the very beginning, the low polymer density polynipam nanoparticle, she actually saw 66% loading efficiency, which is a little surprising, but very good. And in the hybrid particles, she had 76% loading efficiency, which correlates with that after heating, right, after polymerization. And then after we purify away the excess cores, it's down to about 68%. And what she shows is she gets very good controlled release from all three different formulations. So the next question was, could she put the collagen binding peptide, which is called SILY, which is the amino acid sequence at the beginning of it is SILY. Could she conjugate it to the three different particles? She has a small amount of a lipid that has a carboxylic acid on her solid lipid nanoparticle, so she could get some peptide binding to the just the lipid core and better peptide binding to the core shell. And she did a study of how much free peptide does she have to add to conjugate to the surface to get a good amount of collagen binding peptide at the surface. And then she looked at the zeta potential with increasing amounts of peptide on the surface. So zeta potential is how charged the surface of your particle is, essentially. And the more negatively charged the particle, you can imagine the more stable it is in solution, because if they're all really negatively charged, they repel each other. But if they're close to neutral, they can sort of agglomerate together. So they stay pretty negatively charged, but if you add too much of the peptide, certainly to the nanolipid particle, the zeta potential becomes really small and you're at risk of them agglomerating. And then this is the poly-IPM and the hybrid nanoparticle, so she can keep them fairly negatively charged, but really if you stay around this level of conjugation, you're better off, and that binds pretty well to collagen. So she looked at, she put cells on a surface, two different cell types. One are MCF7 cancerous breast cancer cells, the other are T47D breast cancer cells. This cell type has a P53 mutation. We won't go into the details, but what that means is they become resistant to many chemotherapeutics, whereas these do not. So with the nanolipid core, with this degree of collagen binding, so in this area here, you see some binding to the collagen elaborated by the two cell types, same with the poly-IPM nanoparticle, and same with the hybrid nanoparticle. And then she did a study where she took breast cancer cells and grew them in the top of a transwell that has little pores so that small things can migrate through, and put healthy cells at the bottom of the chamber, and then treated the upper chamber with either free paclitaxel or paclitaxel in our nanoparticles, and asked the question, could she locally affect the cancer cells without distally affecting healthy cells? And essentially what she finds is that if she has the nanoparticles that bind to collagen, she can preserve the healthy cells and kill the cancer cells, so it's working locally but not distally. And that's true for both cell types. So that was sort of our in vitro validation that when she goes in vivo, this targeting should help keep the particles local and help kill the tumor and not distal cells. All right, so the schedule said I had an hour. Do I still have a few more minutes? Yeah. Okay. So the conclusions is it's not a long. The rest of it's only like 5 minutes. The conclusions to this part of the story are that we could encapsulate the lipid nanoparticles, we could put the collagen binding peptide on there, and it looks like we can target locally, right? Okay. So I want to remind you though that small molecule went into the poly-IPM nanoparticle without the solid lipid core pretty darn well, which was surprising to me. But the question was, do we need the lipid core at all? So Vanessa Dartora, who was also a PhD student in Luciana's lab and who's a postdoc in my lab now, asked the question, could I just use the poly-IPM particle? And if I were going to just use the poly-IPM particle, can I incorporate that small hydrophobic drug? But also she incorporated a peptide that we had developed in my lab that resensitizes cells that are resistant to chemotherapeutics because of that P53 mutation. Again, we won't go into the biology, just to understand that's why there's this Yara peptide in there now. It's to resensitize cancer cells. There's a small molecule chemotherapeutic called piperotein, and then there's our collagen binding peptide, and it's just the poly-IPM particle now. So she made these particles. These are TEM images of her particles. She looked at the size, either unloaded, loaded with both piperotein and the peptide, the Yara peptide, and then with the collagen binding peptide as well. They stay about the same size. The polydispersity of the particles changes a little bit, so the distribution of size broadens slightly. And the zeta potential is very negative without the collagen binding peptide. It drops to about 10, -10 with the collagen binding peptide, -9. They still stay pretty stable though, so that's good. She looked looked at in vitro drug release, and what she found was that at low pH, like pH 3.5, which at any acidic pH, even at pH 6.5, 5.5, the particles will start to fall apart because the disulfide bond will start to fall apart. We accelerated that by going to 3.5. What you see is the peptide comes out faster at 3.5 than at 7.5, so we'll get better degradation of the particles in a tumor environment where the environment's more acidic than you would in normal tissues. And both the pipe protein and the Yara come out of the particles. She then made little organoids, three-dimensional organoids, in vitro of both the T47D cells and the MCF7 cells, and looked to see how the treatments worked in these little organoids. And what she found in the T47D cells, which have that P53 mutation, that she delivered both pipe protein and the Yara peptide, that she got better killing of the organoids. And this is just the pipe protein, so this is just the chemotherapeutic, and then this is just blank, or this is the chemotherapeutic just in solution, not controlled release. And similar results with the MCF7, but the Yara peptide doesn't resensitize as well, which isn't surprising. Okay, so now the more exciting results, and just the combined therapy with the nanoparticles works a lot better than the free chemotherapeutic. These still collagen binding, I'm not going to go through that. She then went into animals and wanted to see if she injected the particles that bind to collagen, do they stay in the tissue for a long period of time? So what she found was essentially that if she delivers particles that bind to collagen, they stay in the tissue over 100 hours. Okay, and if she delivers free drug, the free drug comes out, or the free, just a free fluorophore, it comes out very quickly. And then she used a breast cancer model where she induces breast cancer in these animals and then treats with nanoparticles at 5, 8, 11 weeks, and then isolated the tissue, used it for histology, as well as looking at the concentration of drug both in the tissue and systemically. And what she finds is that the animal weight is stable, so the treatment doesn't seem to be toxic to the animal. If she induces tumor formation and does not treat with any chemotherapeutic, then there's about a 77-78% incidence of tumor formation. The animals have about 2.5 tumors per animal, and the size of the tumors is about 26 millimeters. If they don't induce tumor formation, not surprisingly, no tumors are formed. And if they treat with the nanoparticle, what they find is there's about 0.15 tumors per animal, and the size of the tumors that you do find is dropped to about 4 to 5 millimeters, so we're dramatically reduced. And if you quantify the amount of drug in the mammary glands, so locally to where they were treated, the concentration is about 35 micrograms, and if you look at concentration in the plasma, it's about 0.5 micrograms. So it is staying essentially local and treating the local tumor rather than affecting the animal systemically. Histology confirms what I just told you, but for time we'll stop. So we're able to make these nanoparticles. We can introduce them intraductally, we can get them to stay locally and release their drug locally, and reduce the size and of tumor burden. We'll stop there again. Folks that worked on this project, funding for this project, and I'll stop there. Happy to take any questions.
Thank you.
Hi, that was an amazing talk. Also, you speak well. I appreciate
Thank you.
how you explain things beginning to end. I didn't understand all of it, but I was wondering for the hard iron chip project, you mentioned briefly that COVID patients get arrhythmias, and then you mentioned that your peptide treatment basically could prevent that. is it that the glycans are messed up or in the heart?
Yeah, so, I didn't talk about this, but when people get really sick, they get systemic low levels of inflammation, and so they lose some of the glycophilics throughout their body. And so, yeah, it is likely then that you're getting some low level of inflammation in the heart, and it's causing these kinds of arrhythmias. And by treating with these kinds of molecules, you can sort of recreate that glycocalyx and reduce that inflammation. But it's one of the problems with sepsis patients too, right? People who are septic get systemic inflammation, right? You can get multiple system organ failure. That's from that overexuberant inflammation.
Do you know if it's like long COVID or just COVID in general?
The arrhythmias, it can happen in any kind of COVID. We were seeing it really, really early on before we even knew that there was long COVID. Yes.
Alco Valentina's praise of your talk. That was really nice to listen to. I had a question about, again, the glycocalyx. We were lucky enough to have Shelly Weinbaum give a talk here a few years back, and he talked about glycocalyx, and it's been in my mind ever since. He was like, I never looked it up after that. But he talked a lot about like the mechanics implication of the glycocalyx and how it's more important for flow at really small capillaries to reduce friction between the red blood cell and the artery. So I was wondering if you could talk a little bit about how the size of the artery and with relation to the cell size, for example, in mouse arteries, but I assume red blood cells are the same size in humans and mice. And then how does that relate to your heart on the chip? Does the glycocalyx, is it recapitulated in vivo and in vitro, like in your isolated PCB? It's
Yeah.
kind of a loaded question.
Yeah, no, and part of it I won't be able to answer because we haven't looked at the glycocalyx mechanics, right? But so we try to design our in vitro system so that it's relatively small. It's not capillary size. It's probably more like a venule size, on the order of a couple hundred microns rather than on the order of like 10 microns, where the red blood cells really have to fold up to fit through. We do try to recapitulate the shear flow that you would see in vivo by modulating the rate of flow, and they do that by the height of the tube that's feeding in. Are we really recreating the glycocalyx with respect to the mechanics? Probably not, right? Because the glycocalyx has all these hair structures, and ours probably sits more like this. It provides sort of a glycan mask over the surface and steric hindrance to being able to interact with that surface, and hopefully stays long enough for the cell to be able to grow its own glycocalyx back. Not sure if that really answered your question.
Yeah, no, definitely, yeah.
Yeah, okay.
Thank you.
Yeah. Oh, go ahead.
Oh, sorry. I have a weird question. Using what you used, what you guys have learned so far with the cancer research, I know you guys are studying breast cancer specifically within the milk ducts. But could you, in theory, like inject those lipids into other parts of the body, let's say like a hard to reach brain tumor? Or would the lipids, could the lipids cause like a stroke? Because then there's that in the brain.
Interesting.
Yeah,
Yeah.
could you apply that
so-
research?
So we could inject them in the bloodstream, and I would be very surprised if they cause a stroke, just because they're so small. They're on the order of maybe 200 nanometers,
Okay.
and they're much more likely to get cleared from the body before they caused any kind of problem. You'd have to get a lot of them binding into
Yeah.
a blood vessel,
A clot,
forming
yeah.
a clot, essentially.
Yeah.
And we've designed them not to do that.
Okay.
We could potentially use them for other tumors as well. I think, the glioblastomas and other brain type are difficult because getting things to cross that blood-brain barrier is challenging, and so we'd have to decorate the surface for something that it would allow it to do that.
Yeah.
But there's certainly other cancers where we could do direct inject or some other things.
Okay. Thank you.
Just had more of like a process question from one of your slides previously where you talked about purification of the pre-Yara nanoparticles. What was can you elaborate on that purification process that those results?
Yeah, so, the first thing that we do is just dialyze, and then we do a form of dialysis, it's, transverse flow filtration, and we do that against a porous membrane that allows particles to stay inside and smaller things to diffuse out through the membrane. Unfortunately, that didn't work well when we had the lipid cores with our shells. The lipid cores stayed within the inner membrane as well. And I thought we were gonna have to do some pretty complex things to purify, but as it turned out, they have different buoyancies, and so we can just use centrifugation
Oh, okay.
to filter out the lipid cores from the NICAM particle. Yeah, so that's what we end up centrifuging.
Thank you.
I think one final question for you. So there's so much work you have done on discovery, drug delivery as well. Can you comment on translational aspects of your research?
Yeah, so-
Experience, you know, what.
So essentially everything we do in the lab now, since I work with Colleen Murphy at Arizona State, we really try, she's a vascular surgeon. We really try to talk with surgeons, understand the medical need, so that we can translate things out. And we have been really successful in translating molecules out, I would say, because we really try to understand the need and design something that will meet that need. I will say the translational piece can be really frustrating because it isn't always the science that kills a molecule. So we haven't translated the nanoparticles out, but a molecule that's very similar to the glycocalyx molecule that I talked to you about binds collagen and can be really interesting in balloon angioplasty, where you actually denude the inside of the blood vessel, you expose collagen. If you could deliver something that would bind to that collagen and look like a glycocalyx, you could potentially improve healing after balloon angioplasty. We designed a molecule like that. We went to New Zealand and Australia, did a clinical trial, it showed it worked really well. And for a whole bunch of business reasons, the molecule died because the company that was developing it had too many things going at the same time. And the VC started fighting about what to do next, and everything fell apart. So, if you start a company, focus. But I would say for all of you, if you really want to do translational work, understand what problem you're trying to solve before you try to solve it, right? And then as you're coming up with ideas on how to solve it, go back and talk to the clinicians, talk to the patients, talk to the nurses, talk to whomever, to make sure your design is reasonable. I can give you one other example on this that isn't from me, but is from one of my former trainees who went through the Stanford biodesign process, and they were in neuroscience that year, and one of the trainees had gone into a surgery where they were putting in essentially a Harrington rod. So first, the patient has scoliosis and they have a really bad curvature in their back. If that curvature isn't corrected when they're younger through multiple different things that they try to do to correct it, they have to put a rod into the back to hold the back straight. And the surgery to do that is really, really long and complicated. There's lots of opportunity for infection and other things. And so the biodesign team wanted to come up with something where you wouldn't have to get to that surgery. And so they designed a product that you could use in younger children, that you put little clips essentially between the vertebrae of the spine to keep it from curving. And they were really excited about it. They went back to talk to the surgeon. He's like, no, I'll never use that because only maybe 30% of these children will ever get to a point where they need a rod. So I'm not going to operate on 100% of them to protect the 30 who need protection. So that idea failed before they even got through the, past the idea phase. So if you're really interested in translation, do your checks and balances along the way.
Thank you. Thank you. One announcement which I should have done at the beginning also. There is a 3-minute thesis competition for the university today at 6 p.m. in Sachs 160. One of your colleagues is presented, Daniela, and there is a competition. I think the people who participate also vote. So engineering participating, I think she's the only one from BME department who's presenting. So please participate. Go there, encourage her. Thank you. Thanks a lot for